see all these. Oh, we've got Czech Republic. Looks like we've got people from all over the world joining us. Thank you for taking time today.
There you go. I'll take the audio off. There you go.
Hi. Hello.
There we go.
Wonderful. All right. Melissa, would you like to wait a couple more minutes before we begin, or?
I think we can go ahead and get started and folks can continue to kind of come on in.
Okay, perfect. Well, thank you everybody for taking time out of your day today. Welcome. It's an honor to gather for the first official online KDM5B virtual research update. Today brings together families, researchers, clinicians, and advocates who share a common purpose to deepen our understanding, strengthen our community, and work towards better outcomes for all those affected by KDM5B related conditions. We are proud to recognize the partnership of the KARES Foundation in today's meeting. KARES is a nonprofit dedicated to advocacy, research, education, and support for KDM5C related disorders, and they may broaden their work to include all KDM5 genes. Their dedication to empowering families and advancing research plays a vital role in moving this community forward. Over the course of our time together, we'll hear from researchers, connect, and build new pathways for progress. Thank you to the researchers for joining us today. Let's begin. We will be starting with a presentation from Dr. Sid Bonka, Professor of Genomic Medicine and Rare Diseases at the University of Manchester.
Thank you, Jordan. Just going to attempt sharing my screen now. Okay, I guess you can see my screen now. Great. Okay, I cannot see you guys, so if there's anything wrong, just shout out. I might not be able to see your hands, etc. So the task that I've been given is to do a genetics 101, which is basically fundamentals of genetic concepts. And sorry, so I work at the University of Manchester. I'm a researcher, but I'm also a clinical geneticist, which means that I see patients with genetic conditions at our hospital here, which is actually on the same road as the university. So we've got a really well-connected university and hospital. Within the hospital, we have a center, Manchester Center for Genomic Medicine, which is actually one of the largest departments of clinical genetics anywhere in the world. And we provide a very comprehensive service to all different subspecialties of clinical genetics. We also, a few years ago, established a dedicated center for rare conditions, which we call Manchester Rare Conditions Center. And part of that center, the mission for that center really is to improve the lives of people and families living with rare conditions. And as part of that center, one of our missions really is to have this engagement and advocacy with patient support groups. So actually, this is really completely at the heart of my research, my clinical work, and all the work that I do. Quite importantly as well, so we are meeting here because we want to learn about the KDM5B disorder, which is involved in something called chromatin or epigenomics. And I'm not going to go through the scientific concepts around that, but suffice to say that there are several dozens of rare conditions which fall under this group. And along with Professor Albert Besson, from whom you will hear later on, we were funded by the Medical Research Council in the UK to set up a dedicated research node, which is for epigenomics of rare disorders, which we called EpiGen Rare. And as part of EpiGen Rare, the remit is to perform more research into these disorders and also have patient engagement and patient education days such as these. So it really, what we're doing today is completely aligned to what our mission is. So, sorry, my screen's got slightly covered with Zoom. Okay, yeah, I can see now. So what I'll do is start from really basics, and hopefully this will give all of us enough background information to understand and follow the presentation that will come afterwards. I must say that some of it might come across as quite basic to those who already know this, but I just wanted to make sure that we start from quite basic so that we're not missing anyone out. So a genome can be imagined as a library. So on your left is a blood cell, and around the red circle, I've drawn a red circle around what's called a nucleus. And nucleus contains our genome, most of our genome. So if you imagine this as a library, it's a source of information for every cell in our body. So this is a picture on the right, is a picture of John Ryland's library, which is part of University of Manchester. And John Ryland was actually the first millionaire of Manchester. And some of you might know Manchester is a very historical industrial town where the Industrial Revolution was one of the first places where Industrial Revolution began. So it's really quite an amazing place. So if you're ever visiting Manchester, I do recommend visiting the John Rylands Library. It's spectacularly beautiful. So having talked about the genome, let's see what the genome consists of. So most human genomes, which is the analogy that I used was libraries, is made of 23 pairs of chromosomes. So you can imagine a library containing lots of books. So you can imagine chromosomes as being those books. So on the picture on your left, you've got these 23 pairs. This is actually a image from an individual who is a male. So you can see a Y and an X chromosome in the picture, whereas if it were from a female, the individual would have had 2 X chromosomes. These chromosomes are arranged as per their sizes, so chromosome 1 being the largest and supposedly chromosome 22 being the smallest. Actually, chromosome 21 is the smallest, but for historical reasons, that's how they were made. And a picture on your left shows a printed out entire volume of a single genome, which exists in our department of genetics. So as you can see, to fit just one person's genome, you need so many different volumes. And the font size in these books is actually only 2 or 4. So it's super, super tiny that you cannot even see or read properly. So our genomes contain a huge, huge amount of information. Let's look at these books or chromosomes in a bit more detail. So you can divide these books into, or chromosomes rather, into arms or bands. And you can think of these as sections of these books. Every chromosome has a short arm, which is denoted by P, and a long arm, which is denoted by Q. So the word P comes from the French petit, which means small. And so every chromosome has a small arm and a long arm. And then each of those arms are divided into these bands. And these bands are given a number or an address as well, you can call them. So for example, this is band 1P36.33, and this is band 1Q32.1. I presume you can see my cursor as well when I'm trying to point out on the screen. Now, this is chromosome 1, or rather, this is a cartoon of chromosome 1. And why have I shown this here? It's because these bands actually contain genes. So again, thinking of genome, chromosome, and bands, and then genes. And this particular band, 1Q32.1, is the location of the gene of interest for this talk, which is KDM5B. And this is the address of this chromosome, of this gene rather, where it starts in this particular chromosome, and this is the address for where it ends. So you can think of these sections containing these genes, which are like chapters of these books. Now, further going down, each chapter is made of pages, as in any books, and genes are made of exons and introns. So they represent the pages in a chapter. And this is a schematic of the KDM5B gene. So each of these squares are what are called exons, and these are what really have the important bits of information for the gene. And then each of these squares are joined by thin lines, which are denoting the introns, which also have information, but relatively their information is could be less important or could be considered to be less important. So this is a schematic of KDM5B. So actually, relatively speaking, it's a big gene. So the chapter sizes or the gene sizes in our genome, they vary quite a lot. So there are some really tiny genes and there are some really big genes. And KDM5B is one of those relatively bigger genes. Now, let's look more deeper into these exons and introns or pages on our chapters or genes. So when you look deeper into it, you will see a sequence of DNA. And you can imagine this as looking like text on a page. So here is a diagram which shows chromosome unravels to make all this DNA, and DNA can be represented as a TGC. And I think most of us would be familiar with this, what's called the double helix configuration for the DNA. And that's actually a screenshot from KDM5B sequence. So KDM5B starts at this ATG, and then it goes on it's pretty long, so I cannot print everything on the page, but to just give an idea, that's what the DNA sequence looks like. And just out of interest, I think some people might find it interesting. I don't know how many of you recognize this, one of the pioneering scientists that we've had. So this is Rosalind Franklin, who actually discovered this double helix structure for the DNA through, and that's called a photograph 51, which is one of the sort of most important discoveries in not just biology, I would say in entire science. So I think it's important to pay homage to these extraordinary scientists as well. So that really provides the basis of what is required to understand how genetic changes can cause genetic disorders or conditions. So having understood what the DNA is, what genes are, and so on, why are they important? So they're important because they make what's called RNA. So KDM5B DNA sequence here is transcribed. So that text is transcribed into RNA. And I'm sure people, especially after COVID and all of that, people are familiar with RNA, but RNA is kind of an intermediate chemical that is synthesized on the basis of the DNA. And this RNA then is translated into what's called a KDM5B protein. And that's really where all the action happens. The DNA is transcribed into a message, which is RNA, which is then translated into a protein, which is the which is the functional unit. So actually gene on its own cannot do anything. It needs to make the protein for the cell to do the right thing that that gene intends to. So gene is just basically like a software or piece of information for the hardware, which is the protein. Now, some changes in the DNA sequence, so you can regard them as spelling mistakes in a text, may result in sorry, there is a typo. Now I'm talking about spelling mistakes, and lo and behold, I've got a spelling mistake on my slide, which is it should have said abnormal protein function. There you go. I think it was honestly unintended. So now let's see some examples of how these abnormal protein function can happen. So let's say you've got a chromosome. So you remember these diagrams of chromosome, and this is a diagram of chromosome 1. And if you've got a deletion, which means basically that part of DNA is completely missing, so what would happen? If there is a deletion at this position 1q34, if you remember what we talked about previously, then this whole bit of DNA for KDM5B would be missing from that chromosome, right? So it's pretty straightforward. And if this bit of DNA is missing, if you remember, the DNA was making RNA, was making protein. So if DNA is not there, the functional part, the protein, will not be there. So that would lead to that person, instead of having 2 copies of KDM5B, having only 1 copy of KDM5B if one copy was missing. And if you remember from our chromosome diagram, we have 23 pairs of chromosomes. Every chromosome of a pair comes from mom, and the other part of that pair comes from your father. That's an example of how a chromosome deletion can cause loss of a function of protein. But there are lots of other ways in which the protein function can be affected. So there's another example which we can call as a truncating mutation. So what does that mean? So now we have to be really careful because the font size is really small. There is no prizes, unfortunately, at this virtual meeting, but can anyone spot that there was a tiny little change? And I can see Melissa nodding. So there was a tiny little change where A changed to a T here. And then sometimes even these very small changes can have quite dramatic consequences. It might seem, oh, just a single spelling change, but what that can do is the transcription might still go on, but when the translation is happening from RNA to protein, that gets impacted, which means that a part of that protein which should have been synthesized cannot be synthesized any longer. So actually, I'm making this relatively simplistic, but there are lots of other biological processes. So it's not as simple as that the protein, half of the protein would be missing. Actually, in most cases, the entire protein would be missing because cell can sense that something is going wrong, and then it just basically dissolves the RNA particle in most cases. But having said that, I think it's fair to say that some of these point mutations can just basically result in the protein not forming correctly or not being there at all. And again, if the protein is not structured normally, or it's not there, or it's missing part of the protein, then it cannot do the function that it was intended to do. So again, that can cause problems downstream. Now, there is a third variety of genetic change, which is called a missense mutation. And again, keep your eyes peeled on the screen. And there's a small little change here from C to G, or G to C. So this is called a missense mutation because rather than this resulting in an entire part of protein missing, it just changes a very tiny little part of the protein, which is denoted here by a star. And sometimes even that tiny little alteration is sufficient for protein not to be able to fold properly or get its right structure or right function. So this is an example of a missense mutation. Now there are dozens of other types of genetic changes, but I've just highlighted the three most common just to get a flavor of what are the kind of changes. So this is in no way completely comprehensive either. So, I mean, we're just talking about proteins, but at the end, what we are concerned about are human beings. So how does this change or loss of protein function have consequences downstream? So what are the consequences of this? So now, if you remember, we talked about chromosome, and KDM5B is on chromosome 1. Everyone has 2 copies of chromosome 1. One is inherited from mom, one is inherited from the father. And then if an individual has a loss of function change in one copy of this gene, or you could have loss of function change in both copies of this gene. So if you've got loss of function in one copy, you could think of it something like a dominant disorder, where the genetic change either might be inherited from a parent or could actually happen for the first time in an individual as well. And that's really important to remember. We inherit all our genetic information from father and mom, but every time you make a sperm or an egg, there are some tiny little genetic changes, and that's how humanity has evolved and grew so different from each other. We are very similar, but yet we are different as well. And those are, to a large extent, due to tiny little genetic changes happening every time when a new generation is created. So a new mutation or a new genetic change can be there, and sometimes it could be inherited from the parent as well. In comparison, when you have both copies with the change, that could cause a recessive disorder in which the genetic changes are usually inherited from both parents, who are thought to be carriers for those conditions. So that's the difference between a dominant and recessive condition. And I think especially for KDM5B, it is a very important concept to remember, because the consequences of recessive KDM5B versus dominant KDM5B are very different from each other. And there's a lot more that we need to understand about this. Now, you would also see that individuals, even with the same disorder, can be quite different from each other. And that's because we're not product of a single genetic change. We all have 20,000 other genes as well. So the consequences of a particular genetic change may depend on the genetic background of an individual, meaning what other changes they have in all the other thousands of genes that we have, but also about the environment, about their lifestyle, and some chance events as well. Sometimes, depending on what infection one has had, or what happens in their lives, or while the cells are dividing. So one must not think that every individual with a particular condition will be exactly the same as everyone else with that condition. So I think I've maybe even overrun my time, but I'll finish with my last slide if I'm allowed. Do I have still a minute or so? Thank you. So this is a summary, just to go over again what we've just discussed. So you remember, so this is a small table here. So on the top is a concept. Then we're talking about what are the possibilities of size of that, and then where KDM5B is relevant and what's the analogy that we used. So genomes are like libraries, and they're huge. So we've got about 10 to the power 9 nucleotide bases there. And then these genome contains books like chromosomes, and they are 23 pairs. KDM5B is located on chromosome 1, which is located on chromosome 1q32.1 band of the human chromosome. And then we actually have more than 20,000 genes. So KDM5B is just one of those genes, which is like a chapter in a book. And we've talked about exons and introns, the DNA sequence. So KDM5B has approximately 4,600 units, bases, those letters that we talked about. And then these variants can come in various shapes and forms, like deletions or truncating changes or missense. which are like spelling mistakes of the text, and they can have either dominant or recessive context. That now we actually discovered the KDM5B disorder, and we proposed that this is a recessive disorder, but there's more work that needs to be done around the dominant disorder. So I'll end there and happy to take any questions now or later on. Thank you for your attention.
Thank you so much for that, Sid. That was very full of information. Thank you. Next, we'll hear from Dr. Astrid Rasmussen. She is a senior program manager at Simon Searchlight.
Sorry, I'm muted. Can you see my slides? Okay, thank you. And I'll just do presenter mode. Okay. So thank you, everyone. Very nice to meet you. I appreciate the opportunity to come and chat with you about our program, Simon Searchlight. I am a physician scientist and currently the director of the program, serving as the right hand, so to say, of our principal investigator, Dr. Wendy Chung. And Simon Searchlight is an international study, 100% based online. And the goal of our study is to create a resource that will bridge the gap between scientists and the families and children affected with complex and rare, ultra-rare genetic neurodevelopmental disorders in such way that we serve as a centralized place where we can collect lots of information in terms of clinical, behavioral, and different aspects of the life of those affected, and optional biospecimens as well, and then distribute them to scientists that are interested in these conditions. We do all this under the auspices of the Simons Foundation. That's where the name comes from. The Simons Foundation is a nonprofit based out of New York City. They have interest in promoting science through the generosity of a single family. And that family sponsors education in math and science and basic science in autism research, which they do in-house. And then they sponsor many other programs like ours to help support those scientific endeavors. It is a budget of 100 million US dollars per year, so it's a very large program and funding effort that goes behind this. And this is our team. Here's Dr. Wendy Chung, as said, our principal investigator. And we are spread through Boston Children's Hospital, the Geisinger Health System in the Pennsylvania area, a team of informatics at the Simons Foundation, and our contractor, Tempus Dynamics. And we have a group of genetic counselors as well. And how we work is that we, as I mentioned, is to shed light on these conditions through building strong partnerships. The goal is to bridge those gaps, to fill in the different needs. And we do that through collecting detailed medical and behavioral histories of the affected participants and also their families. So we try to enroll both parents and at least a couple of unaffected siblings if there's more than one affected child, and all the affected children as well. And we synthesize all the information that we received, and this information can be shared with scientists that apply for it, but we also provide the summaries of the information back to the families and the patient advocacy organizations. the biospecimens and the data we collect is de-identified once we get it. So all private personal information is stripped from these materials, and whatever is shared with other entities or scientists is done so in a coded manner to protect privacy. We also have systems in place to connect researchers and participants. So one way, as I mentioned, is that the scientists can come and request materials from us. But sometimes a scientist has an idea that has not been explored. For example, a new survey, a new questionnaire, a new type of specimen, biological specimen that has not been collected before. And they can come and talk to us, apply for what is called research match, which in my mind is like a online dating service between patients and researchers. So the researcher comes with their best pitch. We put it on our platform. We invite participants that we know have the condition of interest and tell them, John Smith, who's a scientist in, let's say, Australia, is interested in this trait for your community. If you want to participate, let us know, and then we bridge that gap. In many cases, that includes funding the scientist or even creating the instruments online and so on for those studies. So it is more than just doing our standard measures. There are custom options as well. And the ultimate goal is to promote a better understanding of these variants. We study 185 genetic conditions currently, as you can see up here in this little area is KDM5B. We have larger copy number variants included in our study as well. And we currently are working in 7 different languages because we're in an international study. The vast majority, granted, is in English because we're just starting to really launch the other languages. We started with registration and translations of our materials into languages. And this year we'll start rolling out our medical history and surveys in the other languages as well. So that will be coming up hopefully in the first quarter of this year. The phenotype or clinical information we collect is subset in these groups. Their demographic and genetic features. So we start by a background history. Who are you? Where do you live? What is your ethnic ancestry and history? We request a copy of your genetic lab results report so we can make sure it's one of the genes we study. In some cases, if we're in doubt of the quality of the report or if we think this warrants more research because it's a variant of unknown significance, we offer at no cost to do the cascade genetic testing and provide genetic counseling with the results for the family. We collect data on medical history, so general medical history, surgeries, medications, seizures, and previous diagnoses, studies about sleep, and a multitude of different developmental and behavioral scales. Some are publisher-owned, such as the Vineland, for example, and some have been developed in-house but have been validated. And all of them are collected at baseline, and some of them are a longitudinal process. So we want to know how things progress over the lifetime. So we request annual updates on these instruments. We will try to expand to add a few more questionnaires to the longitudinal, including about quality of life. And all this is a dynamic process, so we've been adding things on a regular basis. And the ones we launched this year was that we moved from a medical history survey that was an interview conducted by phone. with a genetic counselor to now doing it online. The reason for this is that we have grown so quickly that we can no longer have the bandwidth of calling everyone. Just to give you a sense of that, we have a little over 9,000 affected participants across all our conditions in the study. So that means that once you put in siblings and healthy family members, we're talking about 20,000 people in the research program. The second thing is that this allows us, sorry, I'll go back one, to also provide pre-filled forms for the longitudinal completion of the form. The annual medical history survey is really, really long. So before, when we tried to do it by phone, it was starting all over each time. Now, each year, participants get their pre-filled form, and all they have to do is update things that have changed in the last year to try to reduce the burden of completion. And the other new survey is called ORCA, which is an Observer Reported Communication Ability score. We have very few people yet that have completed the ORCA, roughly 600 out of our 9,000 participants, because we just started a few months ago. But this one is focused on children with minimal language. To do this process of joining us, there's a participant dashboard. So it's all online. You join and you go through all these different steps. And you will see on your left that there are also rewards. So whether you're in the United States or not, we offer gift cards for survey completion to keep people motivated. And there are different options for each country. We currently have 108 countries participating in Searchlight, so it's super exciting to see that international engagement. And as you can see, we also have ways of tracking what you have done, what you have not completed. We have automated services of text messages, of emails, and so on to help you stay on top of the completion of your surveys. I mentioned biospecimens. This is a voluntary step, so not everyone that joins may want to do this. We are currently limited to collecting blood samples from participants in the US. However, we're also hoping to relaunch abroad this year. We had complications around COVID and then some of the regulations in Europe that make it really complicated, but we think we are working through it. Currently, if you want to donate blood, we send you a blood sample kit to your home free of charge. You just go to any Quest Diagnostics. They draw the blood. They take care of the shipping. You don't need to do anything else. And also launching this year, we'll start offering at-home phlebotomy in the US. So if it's hard to get to a Quest center, we'll start going to your home to do the service. We want the blood for several purposes. The blood can split into several components. After you get your blood drawn, it is spun down in a centrifuge and separated into different components. And in those components, we will have a white pellet, how we call it, that contains all the white blood cells. And from there, we can take out lymphocytes to extract DNA. We can separate all the cells that have single nuclei. And those cells can be, again, used later for DNA extraction, but they can also be converted into stem cells. We call them induced pluripotent stem cells, or iPSCs, and maybe some of the other scientists will talk to you more about it. But these are excellent models to create neurons in a petri dish, and even models of organs in a petri dish, so that we can study the disease, since it affects the brain and we can't just go around biopsying brains. So that's why these samples are very valuable. And what do we do with all this information? I can tell you that so far there are over 120 publications that have used the data generated in Searchlight. We also provide this to sources that aggregate clinical data, such as gene reviews, gene clinics, other portals that families can access to read about the information. Any new information we generate, we provide for free there. Any new variants we find in the samples, we report back to ClinVar so other genetic counselors can use them when assessing variants. And finally, we also want to return results to you as families. So what is the current state of KDM5B in Searchlight? We only have 11 people that have signed up online. 7 have submitted their genetic lab report, and 4 have gotten to the stage of starting to complete surveys, questionnaires, and the medical history. We do not have any biospecimens yet for this community. And our interest in meeting you today is also to see if you're interested in joining our study and having us help you collect this data for your community. Again, everything is at absolutely no cost for you. There are several different platforms or programs that offer this service, but some of them are at some cost. We are fortunate enough to have solid funding and we don't need to charge anything. Scientists are not charged for the materials either. The only thing they're charged for is the shipping cost of the pluripotent stem cells. So a stem cell line costs us $10,000. We ask for $100 to a scientist just to get the cells. So it's actually a pretty good deal in general. Sorry, I don't know why is this going back? Well, okay, sorry. So finally, the summary is that we have 15 years history. We're starting our 16th year under the leadership of Dr. Wendy Chang and the Simons Foundation Autism Research Initiative. It is a multidimensional international program. We share data back. We are currently working on setting up a online portal that will be a self-service option for both the families and the participants and of course researchers to explore our data directly. So not only having to ask for it or work with a scientist so that you can go directly and look at data, of course, always protected with coding so you can't identify the subject. And we are also part of the larger group of Safari, which is the name the Simons Foundation gives to the scientist-facing side. And this is where any scientist interested in the materials can just reach out to Safari Base, and that's where applications are submitted for funding. There's funding for organizing patient meetings. There's funding to sponsor scientists to go to scientific meetings as well. So just wanted to let you know that we're a very comprehensive program and happy to work with you if this is something you're interested in. And we also provide educational materials if that's for interest. And my time's up. Thank you, Melissa, but I'll be happy to listen to questions if there are any afterwards.
Thank you so much for your presentation. That was wonderful. We will now hear from Dr. Julie Seacombe, Professor of Genetics and Neuroscience at Einstein College of Medicine.
Hi everyone. I know a couple of people on this call, not very many. So my name's Julie, and I've been working on KDM5 family proteins for a long time now. And I always like to start with this picture, which is actually, as the person running the lab, I'm not typically the one doing the experiments. I'm the one kind of guiding everything. And these are the wonderful people in my lab that are doing all of the experiments. And as Jordan mentioned, so I started my lab at Albert Einstein College of Medicine in New York in 2009. I've been working on KDM5 family proteins since 2007, so a long time now. And my lab is here, and if you're interested in the latest and greatest, you can look at my lab website. So why am I here? So I'm generally interested in KDM5 family proteins. I've also really been active in the KDM5C community. I met them in much the same way that you guys are meeting each other for the first time. The KDM5C families met each other for the first time in person in 2020, actually in New York. And one of the kind of downstream things that happened kind of in response to that was the formation of the KARES Foundation that you're very familiar with, and both Miriam and Melissa are on the call. And I've been the chair of their scientific advisory board since the foundation was formed. So that's kind of my background. And we've had some amazing talks already that have really introduced some of the concepts that I'll be talking about. And so these are just representatives of, we talked about like the fact that there's a family of these genes in humans. And all of these little lollipops on the lines here just reflect the fact that these are all genetic variants that occur that are associated with people that have some kind of cognitive change, right? And you can see that across KDM5A, KDM5B, and KDM5C, that there are a lot of different changes that can happen that can cause all kinds of neurological disorders. And so really where I'm coming from is like a fundamental interest in understanding all of the KDM5s. But I think that there's actually something to be said about the similarities between the features that you see in those who have changes to KDM5A, KDM5B, and KDM5C. There's actually kind of a lot of things that seems to overlap between the three kind of disorders. And, really what I want to know is understand like what is it about these that's similar, what's different, and that's really what we're aiming to do. And to do that, we kind of use two different systems. So we're definitely my first love is Drosophila. These are a little fruit fly. If you've ever been holding a glass of wine and seen a little teeny tiny fly right there, that's the thing that I work on for the most part. They're an animal model, and they're actually really similar to humans. You would be surprised by the fact that Drosophila, so this is a little fruit fly, 70% of all the genes we have also happen, flies also have those same genes, and they're really doing much the same thing. Flies need all of the same systems that humans do, and all the same genes are needed to form those systems. And across scientific history, Drosophila have actually given us a whole lot of information about kind of this fundamental biology. I guess one of the more recent ones was there was a Nobel Prize awarded for the understanding of the circadian rhythm, which is a 24-hour sleep-wake cycle, all of which was initially discovered in fruit flies. So we actually use both of these systems, but I'll mostly be talking about flies, and because I truly love them the most. And so really the power of flies is they're just a great genetic system. They have basically the same genes, and it really allows us to take a deep dive and connect things that are happening in the nucleus, as Sid mentioned at the beginning, and link that to changes that might be happening in the brain. And I'll talk a little bit about the kinds of things that we can look at in flies. The other thing that we use, which was just mentioned, which was a great introduction from Astrid, which is human iPSCs. So these are cells that are coming from people that we create stem cells from, and that we can then use them to either create specific neurons, subtypes, or we could also create these kind of mini brains to try to understand the relationship between different neurons. And the lucky last thing is that more recently, we've really been getting into something that, again, was just introduced by Astrid, that taking advantage of what we know about the human variants that we have. And we've been really only doing that with KDM5C. And that's because the KARES Foundation has been partnering with RareX. And so we've been trying to look at what is the correlation between these changes to the KDM5C gene and the traits that then present themselves. So we're really interested in, KDM5s. They're actually, these proteins, these genes are actually expressed across many different cell types. But as you see from, looking at your own kids is that, obviously they have really key roles in the brain, but we want to know how is it that these family proteins function across different cell types. We haven't really introduced the fact that it's actually an enzyme, so it actually modifies the way that the DNA is in the cell. And then trying to understand how does this go wrong, right? If we understand how it goes wrong, then maybe we can try to understand how we can best treat that and come up with something that's very targeted that's going to be the best treatment. Okay, so this is where I tell you that although flies are my first love, they're not humans. In humans, there are actually 4 related genes. There were 4. We mostly talk about 3 of them, but flies actually only have 1. And so we'd like to think of this as really being reflective of across functions across all of the KDM5s. And so, yeah, it's going to present with attributes across all of these, which I think is an advantage of our system here. So what we've been doing, and again, it was a beautiful introduction by Sid, is that we've been taking some of these changes, these spelling mistakes, and so by putting them into flies and then saying and asking, if we just have this one specific change, maybe that will tell us something about the way that the protein, what goes wrong with the protein function. And what we think is the power of this is the fact that the specific amino acid that's changed here, so there's one specific spelling mistake, actually occurs in a region that is the same across all of the KDM5s in humans. So we think this is going to be telling us broadly what's going wrong with KDM5s. So why are we using flies? Actually, I'm not going to talk about that. So we use flies because it enables us to connect things that are going on in the nucleus with behaviors. You might not think of flies as being particularly sophisticated, but they actually are surprisingly sophisticated. They learn, they have a learning and memory system very much like ours. They have social behaviors, they interact with each other, they have favorite flies that they like to interact with. We can model things like seizures, which is a common feature across all of the KDM5s. And we can also look at things, what's happening with the axons and how they function. So again, I like to emphasize the fact that these are the really amazing people in my lab that are really pioneering these studies and trying to understand what's changed about these behaviors, and then trying to understand what's going wrong to cause these changes in behavior. And I just wanted to, because I'm assuming that many of you are not particularly familiar with flies, I just wanted to show you the kinds of stuff that we do. So for example, learning and memory in flies, they have a really robust learning and memory, and it's, again, all of the same molecules that we use to learn and remember stuff, flies do it, it's on a different time scale. And the way that we can test that is much like us, like we have a really strong sense of smell and associating with smell, right? That smell of the dentist is something that I think that we all remember, right? It's like you smell the dentist and you're like, that's the dentist smell. So flies do the same thing, and we can associate a particular odor with a reward, and then what the flies will do is actually remember that. So for them, short-term memory is 5 minutes, and long-term memory is a day. That's not the same scale as us, but 24 hours, about a day in the life of a fly is about a year in the life of a human being. And then you can test that. You can ask, do you remember which odor was associated with the reward? And we've done this with KDM5 flies, and we see that when you change KDM5, you affect the ability of the flies to remember. The other thing that we can do, and another feature that I don't know how prevalent it is in KDM5B, and I'd love to know more about it, is certainly for KDM5C, there's a lot of changes to the way that the kids move. And they don't necessarily have the best coordination, for example. And again, we can actually use the flies to quantify this. And as you can see, the video playing right there is that we can just track the flies. We can track all of the flies, and we can quantify exactly how far they travel, how well they travel, do they fall over, like all these kinds of metrics. And all the dots over here just tell you that KDM5 is actually required for the flies to walk normally. The other thing that's actually the most recent model in my lab is actually seizures, which is pretty common amongst all of the KDM5s. And again, you might be wondering, is this really like, it's a fly? Does it really have a seizure? So I just wanted to show you what it looks like. So flies move around like all of we do, and it's like what we can do is if we heat up the flies a little bit, we can induce them to have these seizures. And so they look very much like a tonic-clonic seizure that happens in people, and you can see poor thing is still having a seizure right now. But what they do when we take away KDM5 from neurons, they actually have these seizures and they right themselves and are perfectly fine afterwards. The lucky last thing that I'm going to talk about is sleep. And again, I don't know how KDM5B affects sleep behavior in humans. Certainly, this is something that comes up very frequently in the KDM5C community, is like an altered sleep. Kids don't necessarily sleep at the right times, right? They don't necessarily want to sleep at night. They're more likely to sleep at kind of inappropriate times, I guess. And this is something that's really famously been studied in flies. And what we do is we just quantify their movement. And if the flies are not moving, basically they're asleep. And so we have these little tubes here, like all of these tubes here actually house a single fly. And there are a bunch of laser beams going across those. And we can see when a fly crosses, it breaks the laser beam. And so we know it's moved. And so what we see is, this is probably a little bit too much detail, but what you can see is like the bit shaded there is actually the nighttime. And what you can see is right when dawn and dusk happens, is the graph kind of dips down. And what that means is that flies are most active at dawn and dusk. And so there are fewer flies asleep at those times. And what we can see is that in typical flies have this really kind of U-shaped kind of pattern here. And, oh, whoops. When we look at some of our variants here, what we see, for example, some of the more dramatic ones here, is that some of these flies, they just sleep at any old time of day. Their total sleep is actually about the same as typical flies, but they're sleeping at inappropriate times. And so we're trying to now try to mimic things that could maybe restore this cycle. And so get them to sleep at the right times and see how that can then impact, things like cognition and stuff like this. And this to me is the beauty of flies. We can start to really look at all these things in like really minute detail, and then also go back and then test some of these things in things like the iPSC induced neurons. But honestly, the cells in a dish are not the same as an animal. Like there's really something to be said for having a whole animal model in all of this. All right, so this is where we're at. We try to combine our studies in the flies, and then we try to see whether those things are conserved in some of our iPSC models. And we're trying to understand what is going wrong. And with the goal that if we understand what goes wrong, then we can try to fix what goes wrong and see how that can see what happens to some of these phenotypes, some of these kind of traits that we see in the flies. And that's it. That's kind of a taste of my lab. If you're ever in New York, we'd love to have people come visit. Again, like these are the people who did the work. I feel very fortunate to have just a wonderful group of people in my lab. And of course, the funding for my lab that mostly comes from the NIH, which we all need to pray for. And so I'm happy to take questions at the end, but that's it. Thank you.
Thank you very much for sharing with us. All right, so our last researcher presenting today will be Dr. Albert Besson, Professor of Neuroscience and Epigenetics at the University of Exeter.
Great. Thank you. Can you see my slides? You can hear me okay? Brilliant. Thank you very much. So I'm really following on from what both Sid and Julie had told you about. So my lab, very similar to Julie, are really interested in trying to understand the function of genes. We don't work on fruit flies. We use mice as a model system. For very similar reasons, we can very easily manipulate the genes in mice. And because mice are mammals, so as Julia told you, fruit flies have one KDM5, but mice have all of the 4 different KDM5 genes that we as humans have. for some reason, I'm not moving forward. Looks like I need my cursor. So just going back to what Sid had already told you about, he's told you that all of our DNA is contained within the nucleus, and the DNA is essentially wrapped around these what we call nucleosomal particles that allow the DNA to be compacted into the chromatin. And when we look at the can be compacted into the nucleus, if we look at the sort of structure of these, we see that we can have parts of our chromatin, parts of our DNA that are very condensed. They all sort of compacted, and parts of the chromatin that's open. And what this essentially means functionally is that genes that are sort of within these open chromatin regions can be read by the cell, and the genes can be turned on. Whereas genes that are contained here in the more compacted chromatin state are being turned off. So how does the cell know what genes to turn on and what genes to turn off? So I sort of think about it a little bit as a traffic light system. And I'm afraid there we go, it's not working. So what this cell does is it can actually label chromatin with these little marks that can work almost like a traffic light system. So the parts of the chromatin that's marked by these little red lights, the cell knows, okay, I've got to turn this chromatin off, and the genes that are within this part of the DNA is to be turned off. Whereas those that are labeled in green are kind of kept open and the genes are being turned on. So these are, where the gene will be transcribed into RNA and eventually protein being made. So why am I telling you this? I'm telling you this because Siddha told you that we really need to look at the protein. KDM5B protein is where the action is. We need to understand how different mutations affect the function of the protein to understand how it results in these different conditions. So what KDM5B does is actually, it's an enzyme, as Siddha told you, that removes these little green marks, that remove the go signals. So that's the sort of normal function of KDM5B. So what we think, in most cases, if we have mutations or different genetic events that reduce the function of KDM5B, we should be adding on more of these green signals, and some important genes may be overexpressed. We might be making too much of that particular gene. So that's kind of what we're essentially setting out to test in some of our model systems. And I'll show you some data a little bit later on to show that we have evidence that that is actually what's happening. So, as I said, we're really interested in trying to understand the function of KDM5B in the brain using mouse models. And when we started working on KDM5B in about 2019, there was really very little known about it, almost nothing. We knew that KDM5 variance was associated with autism, but that was really about it. We now know that actually it's not just associated with autism, but also with ADHD, with learning difficulties. And what we sort of set out to do was just to say, well, let's see if we can understand what the function of KDM5B in the mouse is, and maybe we can study some of the key symptoms that's associated with KDM5B mutations in mice. And if we've got a good model system like a mouse, and we understand the mechanisms, maybe we can start thinking about identifying and potentially developing some treatments for some of the most debilitating symptoms. So we were lucky enough to be able to get hold of a KDM5B mouse model. So this is really an experimental mouse model to study KDM5B function. I really want to make the point that it is not necessarily a representative of the human condition. So we've not introduced any of the sort of human mutations that we know are associated with symptoms. This is a large deletion in the gene, and both copies of the gene is affected in these mouse that we're studying. So we're really only using these mice to understand what the function of KDM5B is. But as I said, hopefully we can learn a lot more about the function of KDM5B using this mouse model, and hopefully it can, take us forward. in that. So in the next few slides, I'm just going to show you some of the effects, so very similar to what Julia told you about the flies, what we've learned about KDM5B and how mutation in KDM5B can affect the mouse. So the first thing we noticed that actually these KDM5B mice were grew a little bit slower, and they ended up being a little bit smaller. They had a sort of lower body weight than their sort of wild type normal counterparts. So again, as Julia said, we know so little about KDM5B. It'll be really important for us to know whether this is something that is also seen in individuals with KDM5B mutations. So that's just showing again the data. This is now just looking at adult mice. Each of these little dots represent an individual mouse. And you can see in general the KDM5B mice here in these red bars are smaller. They weigh less than the wild types. We also measured their brain size just by weighing the brains, and the brains were pretty much identical. But because the actual mice were smaller, the relative size of the brains were actually bigger. And we've seen that. We've done some MRI studies in these KDM5B mice. And again, their brain size seems to be slightly bigger. So again, that's something that we'd be very interested to know whether that also applies to humans with KDM5B mutations. We've not seen any other sort of major structural brain changes. So some of you might be aware in some of these neurodevelopmental conditions, you can actually see structural brain defects. So you can see parts of the brain are significantly smaller, or you may have, axon tracts like corpus callosum being missing. We've not seen anything like that in our mouse models. Of course, that doesn't mean that can't occur, but so far we've not seen that. So next, thinking about very broadly, conditions like ADHD and learning difficulties that are associated with KDM5B, we can very easily see whether our mice have any changes in activity and whether they have learning difficulties. So we've done that. To just look at activity, we essentially just place the mice in a what we call an open field. It's basically just a box that you can see here with a camera above, and we can record their movement. And one of the things that we saw, again here you can see in the colored graphs, that actually our KDM5B mice were hyperactive. So they did seem to move much further when they placed in this field. We can again look at learning and memory. So this is just a test where the mice are placed in an arena. We've got two identical objects there. We train them, they learn what these objects are, and the next day we just move one of the objects to see if they can remember the position. And it may be a little bit of a complicated graph, but essentially anything above the line here shows that the mice have learned. So these are the wild type mice, and you can see that most of them have learned, but our mutants really struggle. Some mice can still learn, but the majority of them really are not able to learn this task. So we think we can model some aspects of learning difficulties using this mouse model. So what about autism? So obviously KDM5B has been associated with autism. So we can look at some of the core features like repetitive behaviors and social behaviors. In terms of repetitive behaviors, one of the simple tasks we do is we just put a mouse in a cage with some sawdust, and mice that tend to be very repetitive, they tend to just start digging away in the sawdust much faster than other mice. We place marbles on top, and then we can just look after a certain amount of time how many marbles have been buried as an indication of how much the mice are digging. And we saw a very clear effect here in the mutant mice that they were burying these marbles much faster, so they had a real clear sort of repetitive digging behavior. Social behaviors, one of our favorite tests to look at sort of development of socio-communicative behaviors in pups, is that we can remove the pups from the mother during early postnatal development just for a few minutes. And the mice, like any other, like a human child, when removed from a mother, the mice tend to just shout out to the mother. So they vocalize, but they do this in the ultrasonic vocalization range. So we can't hear it, but we've got a special microphone and we can record that. And we can just quantify that. So you can see here, these are mice that are little baby mice that are 2 days, 4, 6, 8, or 12 days old. And we can count how often they vocalize. And you can see they really vocalize a lot when they're sort of around 8 days old. So that's control mice. When we looked in the mutants, the KDM5B mutants, we saw that they really vocalized much less. They had a clear deficit in this behavior. So this is a standard test that people use quite a lot to look at autism-like behaviors in mice. So to try and understand the mechanism, so I've told you earlier on that the KDM5B protein is really important in controlling gene expression. So we can look at the expression of all of the genes within the brain. So each of these little lines here represent a gene. And all of these genes that are here highlighted in yellow are genes that are expressed at higher levels than they should be. So they're too highly expressed. So that fits with what we think KDM5B is doing. So when we take KDM5B away, most of the genes, many of the genes, are overexpressed. So by looking at these genes, we think we can maybe, get a better understanding of how KDM5B might cause, some KDM5B mutations might cause neurodevelopmental disorders. One of the genes that were upregulated was a gene called GRIN2D. And this is an interesting gene because it encodes one of the receptors that the cells in the brain use to communicate with each other. So we think that there may be some hyperactivation, like an overexcited signaling between neurons in the brain. And this was really interesting because there are actually some inhibitors, some drugs that have been developed that can target this specific mechanism. So we took our KDM5B mutant mice, we treated them with this drug, and we could show that there was actually an increase, a sort of improvement in their socio-communicative behaviors. So this just shows that we can use these mice to understand and identify some key mechanisms, and perhaps we can start identifying some drugs that might show some benefit in certain contexts. But obviously very, very important that I need to make the following point. These are very preliminary data. It's an experimental mouse model. We don't know to what extent this will apply to humans, and a lot more research needs to be done to understand whether this will apply to humans, and of course, if any of these kind of treatments will be safe and effective. And most importantly, whether this is something that is relevant and important for the community and for individuals with KDM5B mutations. And I'll come back to that just in my final slide after this, just to acknowledge the people who were involved. I started this when I was still at King's College in London, and the work was taken on by a really talented postdoc in the lab, Letty, that you can see over here. She was helped by a PhD student, Shale here, and now that I'm in Exeter, a lot of this work is being done by Josie over here, and we have lots of other really fantastic collaborations. So finally, and I think really just following on from what Astrid had said, we really need your help. We are kind of flying in the dark to some extent. We have these models, we're trying to understand what KDM5B does, but we know so little about how KDM5B affects individuals. So we need to understand what the most significant symptoms and diagnoses are that are associated with KDM5B. What are the symptoms that really cause most difficulties and would be priorities for research for individuals and for families? We were planning to put a questionnaire together to kind of share with the group to just help us understand that. But obviously, as Astrid had said, the Simon Searchlight is trying to kind of do that and facilitate that. So I guess as a community, you guys can decide what will be the sort of best way of going forward with that. But we really need a lot more information from you that will hopefully be helpful. And that's all. Thank you very much.
Thank you again to all of our researchers today. This was very informative, so thank you for being part of this community and supporting our families. Now, I'm not sure, Melissa, do we have time to take questions or?
I mean, I think we're in a unique position to have the researchers available, and we can always schedule family meetings, as you guys wish. So I think we should open the floor if there are any burning questions for our researchers while they're still here on the call. I have a question, actually. In just talking with KDM5B families, it seems like those with two variants are considered kind of pathogenic by the science, but then there's also a good representation of folks in the group that have one variant that seem to have some developmental impact. Can anybody speak to that on if that's been modeled anywhere?
I'm happy to take that question if that helps. But yeah, please, Julie, Albert, to chime in. So officially, definitely so far. So OMIM is a website which catalogs all the human genetic disorders, and it only catalogs the recessive form. So both copies, where both copies are affected. It has been shown that having one change or change in one copy is likely to increase the chances of neurodevelopmental problems, but not everyone who has a single change has neurodevelopmental problems, which are obvious. And sometimes perhaps more objective testing may reveal something which is quite subtle. So right now the thinking is that the recessive form, two copies, is definitely a genetic disorder, whereas the dominant form, one copy change, is could be thought of more as a susceptibility factor rather than a bona fide genetic condition. And I don't know if Albert and Julie, you want to, do you agree or do you have difference in opinion?
Yeah, I think that's something we're very interested in the mice. So that's definitely my understanding from the human population as well, is that there are individuals with, maybe parents even that have, dominant or, one copy of a KDM5B affected, and they're not really affected. So it's one of those genes where it really is just the risk factors. So we've not tried extensively to model that in our mice. We're really interested in doing that. When we just look at the expression of genes, similar to what I've told you in our homozygous mouse model, we do see lots of changes in genes. So certainly losing one copy can affect gene expression, but we don't know what the effects are on sort of neurodevelopmental outcomes. So we're hoping to study that in the future.
And this may be one of those instances in which a scientist could partner with the Simons Foundation and start a study trying to recruit specifically those carriers that have only one mutant copy of the gene to do a study into it. So when I was saying that we try to play matchmaker, so to say, between scientists and participants, maybe you can identify a scientist that would want to actually do this in humans. Just collect the data, see what the patterns are, and we could be a facilitator in collecting the phenotype data, putting you in contact, and even doing the biospecimen collection and distribution and so on. So think about it as one possibility.
I don't know how controversial can I be. Is that helpful or not very helpful? So no, I won't be. I get the message. But I think I would say that one has to be super, super careful in conducting these studies. So there's something called ascertainment bias that we must avoid when we are trying to interpret cohort studies like these. But I completely agree. Having contributing to Simon Searchlight would be a wonderful, wonderful effort.
Are there any other questions from the group? Seems like there were a few in the chat, and I will definitely go back and collect those up and send them out to our researchers so we can get some feedback for everyone. What we'd like to do now, Jordan and I were kind of chatting in the back channel about, I know there's a few KDM5C parents on here, there's some researchers on here, but I don't think the group is too big for us to do like large group introductions of the KDM5B family. So I'm going to stop recording at this point.